Oh, what is up, everybody? Okay, so this is the molecular genetics unit. This is the second to last unit that you guys have to worry about for this half of the year. Unit 5, total of 6 units to worry about on that test on December 13th. For this unit, we literally just went over it. So these are all of the expectations, all pretty simple stuff. This is the stuff we wrapped up right before Thanksgiving break. So let's just jump right into it. So we're going to talk about nucleotides and nucleic acids. So for nucleotides, you need to be able to identify those three components. So those of you that might have looked at the flashcards, you see you got your phosphate group, your sugar, and your nitrogenous base. The nitrogenous base can change depending. So this right here, when we throw those letters out there like A, G, C, U, T, when we're doing our transcription, translation, all that, those are the nitrogenous bases. Those are what we're talking about when we are talking about the nitrogenous base. Okay. So you need to be able to identify these on a diagram similarly to what was on y'all's test for this unit. And I've got a picture pulled up here for you guys. Now this looks like a hot mess, but you guys actually already know everything that's going on in this picture. So in this picture, we have our DNA. Here, what does this look like right here? That little P right there with all those O's looks kind of similar to what we've got going on right here. That's because the thing that connects nucleotides together in a DNA or RNA sequence are these phosphates. Those are what are connecting those together, as well as the nitrogenous bases, very weakly being held together from opposite ends. So here we've got our phosphate, we've got our sugar, and we have our nitrogenous base, okay? Just like we have in here, we've got our phosphate, our sugar, and our nitrogenous base. All right, so that is what they expect you to be able to do. They expect you to be able to look at this mess or something similar and to be able to say, yep, that's phosphate, yep, that's a sugar, yep, that's a nitrogenous base. Okay? Next, you are expected to know the two types of nucleic acids are found, the functions of each, and be able to identify each in a diagram. So this is also in here, and I threw in a nice little chart to help you guys remember. So the nucleic acids are actually named after the type of sugar that makes up their backbone. So for DNA, it stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. I don't think you guys need to remember that. You will be expected to know that DNA is a standard double strand. What we mean by that is everybody here's seen a picture of DNA. This right here is what they mean when we say double strand. It kind of looks like a twisted ladder. All right. Additionally, you need to know that thymine is unique to DNA and uracil is unique to RNA. So for example, whenever you guys are doing transcription, you're transcribing DNA to RNA, remember how every time you had an adenine, you had to translate it to a uracil. So AU, so remember C, G, G, C, and then A, U, okay? But every time you had a thymine, you would translate it to A. We're going to go back over that, I think, in a completely separate video, codons, because that can get pretty tricky, but as long as you follow the steps, you'll be fine. You are expected to know that DNA in a eukaryotic cell is only in the nucleus. It is nowhere else in the cell. It is only in the nucleus. The RNA is in the nucleus and outside of the nucleus, within the cell's cytoplasm in many different forms. All right, so you're expected to know their functions, where they're located, and the unique nitrogenous bases. So thymine, so the T is only in DNA. The U is only in RNA, okay? So the functions of RNA are they control the assembly of amino acids into proteins. So let's take a look here at what's going on. So we have our messenger RNA. This is what we got as a result of transcription, okay? So when the DNA was unwound, tried to match up with base pairs, okay, this is what we got. This is the result of transcription, okay? And then, right now we're going through translation where the ribosome, that's why it's called rRNA, the ribosomal RNA, is bound, all right, running along, reading this, binding to the transfer RNA, because these little dots right here are amino acids. And those dots come together to form proteins. So that's how we make proteins. We have our messenger RNA, our ribosomal RNA, and our transfer RNA all working together to bind together amino acids to form the proteins that we need. The function of DNA is to store all of those instructions for us, all right? And then I think after that, yep, semi-conservative DNA replication, being able to identify the stages on a diagram. So when DNA was first discovered, many different scientists tried to determine the way in which it could have possibly duplicated itself. And there were 3 models that were generally accepted and needed to be proven. The first one was the conservative model. So this model, according to this model, we started with a parent and we just ended up with an exact clone, okay? So the original strands both stayed together and we had an exact clone. That's the conservative model. This was proven to be false. The dispersive model, this one, in my opinion, is the dumbest of the 3. I don't know why this was even considered, but hindsight's 2020. Basically, this one was like, hey man, this whole thing, it just shreds up and then it fills the in-betweens. No, this is just a mess. This was proven false. This is what we ended up going with and understanding and found to be true. Okay? Each strand separates, makes complementary base pairs. All right? So this is called the semi-conservative model, and this is how DNA replicates. These other 2 are not true. Okay, these are just the proposed models when we were still figuring out how the heck these things replicated themselves. So in the semi-conservative model, each strand of DNA separates and then makes its own complementary strand, okay? So that's how the semi-conservative model works. You guys need to be able to understand how to read a codon chart. Me personally, I prefer that circular chart that I've been showing you guys, but with this, so see how it says first base? So you need to look for your first letter. So let's say your first letter is an A, and your second letter is a C. Your second letter, you can see it up here. So A, and then C, and then over here it'll give you your third letter, A. It's 3, okay? I'm going to make a whole separate video on that stuff. And then they just want you to know your general mutations. These are pretty simple. So in a chromosomal deletion, part of the chromosome is quite literally deleted, okay? We had A, B, C, D, E, F, now we just have A, C, D, E, F. Now we're just missing an entire portion. So this is a loss of a portion of a chromosome, and this can result in many defects in humans. A lot of the time, it just results in an illegitimate, unpassable fetus that never develops. Then we have chromosomal inversion. So remember, inverse means sort of like in reverse, okay, or opposite. So notice how we have A, B, C, D, E, F. Now we have that B and that C. They switch places, all right? So in chromosomal inversion, the C and the B became inversed. In chromosomal addition, just what it sounds like, literally an addition of DNA, all right? So deletion, we're losing some. Inversion, it's switching. Addition, we're adding some on. Now, this part is where I see some potential confusion occurring. So here we have types of mutations that can occur. So let's take a look at a nonsense mutation. What a nonsense mutation is, it introduces a premature stop codon on mRNA. And what I mean by premature stop codon is not every 3-letter combination codes for an amino acid, because we need to stop eventually. Some 3 nitrogenous base combinations just tell us to stop producing or to stop building the protein. We're finished. So once we get our amino acids lined up and the protein's ready, we get to a stop codon. So for example, the letter U, A, and G are a stop codon. All right. So if we had a U and an A and a C, we get tyrosine. But if we had a nonsense mutation that turned that C into a G, instead of tyrosine, we'd have a stop codon instead. So that would be a nonsense mutation. It's a mutation that would have us making a stop codon too early. All right. So that's what a nonsense mutation is. A missense mutation is when we get a different thing entirely. All right, so in a missense mutation, so for example, in this first one, we had an A or a TCC resulting in an arginine, but in this missense mutation, we changed it from a C to a G. So there was some kind of mistake that occurred that caused this C to turn into a G. And now we end up with a completely different amino acid than what we were intending for. So these are the 3 mutations that they expect you to be able to identify. So they're going to point, they're going to tell you like, so what happened here? So in a missense, we get a completely different amino acid just by changing a single letter. In a nonsense, we get a stop codon instead of an amino acid. In a silent mutation, nothing changes. We end up with the same amino acid because a lot of the time, well, enough of the time, just changing that 3rd letter may not necessarily change anything about what we end up with. So for example, we changed it from TTC to TTT, okay? And it's still a lysine. It's still what we end up with. Let's go over and look at our chart. So for example, if you have GC, so if your first 2 letters of your codon are GC, it doesn't matter what that 4th or that 3rd letter is, you're always going to get alanine. So alanine, one of the easiest silent mutations to miss, because GC, it doesn't matter if your 3rd letter is a U, a C, an A, or a G, you're always going to end up with alanine. All right, so that's what a silent mutation is. Just because we changed a letter does not mean that the amino acid is going to be affected at all. All right, so those are the types of mutations. So let's identify each. Okay, yeah, we need to be able to identify those steps of DNA replication. So in DNA replication, again, this is not transcription translation. That's when we are generating messenger RNA from a segment of DNA. DNA replication is when we're duplicating our DNA. Remember when we were talking about mitosis and meiosis, how we were doubling our chromosomes to get ready for division? That's what this is. So in DNA replication, first is the initiation. Okay, we have to unwind our DNA because, as it turns out, when we're messing with a twisty ladder like this, it's kind of hard to work with when it's in this structure. So the first step is initiation, where we start to unwind it. Okay, then there is elongation. That's when we start connecting the appropriate nucleotides to the DNA. You don't need to know all of the complicated stuff that's going on in these 3 steps. These are the simplest images I could find to put on here. Really, all you need to know is that during initiation, we're unwinding. During elongation, that's the process of actually, we're starting to actually duplicate our DNA. This gets pretty complicated, but you don't need to know it in vivid detail. You just need to know initiation, we're unwinding. Elongation, we're adding on those nucleotides. Termination, we're done, we're finished, all right? It's over, everybody go home. All right, so that's initiation, elongation, and termination of DNA replication. All right, and that is, here, let's see. Yep, identifying each stage, transcription, translation, including, so we already talked about this, the steps of, this is translation. So you got your messenger RNA, your ribosomal RNA, and your transfer RNA. And again, like we talked about earlier, the ribosome reading the messenger RNA, guiding the transfer RNA to form these proteins from these amino acids. Okay? You'll be able to use a codon chart. Now, there is a question every year, and it always gets students. All right? So I'm going to share it with you right now. So, this first, what amino acid comes from the DNA sequence CGA? Or what amino acid comes from the messenger RNA sequence CGA? One of these is a little bit more difficult than the other, but not by much. You need to know that when we're looking for an amino acid sequence, you're only getting an amino acid sequence by directly reading from the messenger RNA, okay? The DNA is not what is generating your amino acid. So for example, look at this. We're not getting our amino acids, okay, to form proteins from a strand of DNA. This is RNA, okay? So if they ask you what amino acid comes from the DNA sequence CGA, well, first you have to transcribe it. So what's the matching base pair for C? It's G. What's the matching base pair for G? It's C. And what's the matching base pair for A? This one's trickier. It's U. So G, C, U. So first we have to find the matching pairs for this because it's DNA. So we need to generate our messenger RNA sequence, which is GCU. So we got our G, we got our C. All right, so GCU, alanine. All right. Now in this one, we already have our mRNA sequence, so we don't need to transcribe it. We don't need to find the matching letters because this is already messenger RNA. Okay? DNA just gives those directions to make the appropriate messenger RNA sequence. mRNA is what we use to actually generate, okay, the codon for that specific amino acid. So we've got because what a student does every year is they look at this and they go, all right, well now I need to transcribe it, so I'm gonna write G, C, U, no, this is already transcribed, it's already messenger RNA, you don't need to. DNA, yes, you need to find your matching pairs because this is DNA, it is not RNA, okay? So CGA, so we go C, then G, then A, so that's arginine, all right? These are very different. These are not the same question. What amino acid comes from the DNA sequence CGA? Okay, alanine. What amino acid comes from the mRNA sequence CGA? Arginine, all right? Distinctly different from one another. So after that, I think that basically gets into all of it, and articulate how these mutations may or may not affect the phenotype, and we already talked about that. The genotype is the physical genetic sequence. The phenotype is what we end up seeing. So again, just because we changed a single letter does not always guarantee that we're really going to change anything, because that could just be a silent mutation. So just because we changed the C to a T, it would still end up with lysine. It didn't matter anyway. But yeah, that's pretty much it for that. The last bit of information they wanted you to be aware of, I might make a separate. I'm gonna make a separate video for that. It's some cool stuff that's very medically relevant, and this video's already going on long enough, and I know y'all have short attention spans, so we'll call it a night. And I'm tired, so take it easy.